Let us begin with meiosis 1, but let us first quickly revise homologous chromosomes and sister chromatids. Homologous chromosomes are a pair of chromosomes inherited from each of the parent, that is mother and father. And we even saw that these homologous chromosomes were similar in length, gene position and centromere location. Whereas sister chromatids are formed after DNA replication. When the cell undergoes cell division, initially the genetic material increases and we get sister chromatids out of homologous chromosomes. So here these two pink colored which you are seeing are sister chromatids that have been formed after DNA replication. Here one more you can see behind, that is purple in color. They are also a pair of sister chromatids. So I hope now that the difference between homologous chromosomes and sister chromatids is clear and that we can proceed. The first one is prophase 1. Prophase 1 of meiosis is divided into 5 stages. This information you will not find in the textbook, but you are expected to know. So the prophase 1 of meiosis 1 is divided into 5 stages. These are, the very first one is called leptotine. What happens during leptotine stage? The chromosomes begin to condense and become short and thick. So here the condensation of the chromosomes has started. We know that initially the genetic material is highly dense and it comprises only of thin thread-like chromosomes. But slowly they begin to condense, that is they become short and thick. So this is what we observe in the very first stage of prophase 1, that is leptotine. The second stage is called zygotine. During zygotine stage, we find that the duplicated sister chromatids remain fused at the centromere throughout the meiosis 1. So here we find that the chromosomes, homologous chromosomes have been converted into sister chromatids after DNA replication, and these duplicated, that is doubled, now sister chromatids remain fused at the centromere and they are going to remain fused throughout meiosis 1. Synapsis begins with synaptonemal complex forming between homologous chromosomes. Now these homologous chromosomes undergo a process which is called as synapsis. So in synapsis, it is just that the pairing of the two homologous chromosomes is going to take place. So here you can see that both the homologous chromosomes have come in contact with one another. They have got connected to one another. So this is what we call it as synapsis. That is, synapsis is the pairing of the two homologous chromosomes. And this is what this connection between the two is called as synaptonemal complex. So synapsis begins with synaptonemal complex forming between homologous chromosomes. Now what is the function of this synapsis? Why synapsis takes place? So later we will see that the genetic material is going to get exchanged, and this is because of which we find genetic diversity and variety is observed in new organisms that are formed. So for the exchange of genetic material, the first stage that takes place is the synapsis, or the connection between the homologous chromosomes. So these are the two stages. The remaining 3 stages we will see in the next slide. The third stage of prophase 1 is called as Pachitine. Number of important changes are going to take place during the Pachitine stage. Firstly, crossing over of genetic material occurs between non-sister chromatids. So you can see here this the two of them over here. These are non-sister chromatids. We cannot call this chromosome and this chromosome as sister chromatids. So these will be called as non-sister chromatids. So there is going to be exchange of genetic material between these two non-sister chromatids. This is called as recombination or crossing over. Thus, sister chromatids do not remain identical to each other. So now let us try and understand what changes are going to take place during crossing over. So synapsis has already occurred. The two sister chromatids are already in contact with one another. And finally, there is exchange of genetic material between the two non-sister chromatids. So here it is clearly visible that how the genetic material from one sister chromatid is transferred to another sister chromatid. So there is exchange of genetic material between the two non-sister chromatids. As a result, now the sister chromatids will not remain identical to each other. Now here it's very clearly visible that the sister chromatids are now not going to remain identical to each other. Initially you would see that they were exactly identical to one another, but now they are not identical to one another. This is why no two siblings except for twins are identical. So now you understand that due to the exchange of the genetic material, we find that no two individuals except the twins are going to be identical. The crossing over is called as chiasma or chiasmata. So this crossing over is called as chiasma. It is also called as chiasmata, and I hope now this is clear. That is what is Pachitine. In Pachitine stage, the crossing over or the exchange of genetic material is going to take place, and this is called as recombination or crossing over. So 1 mark question again is asked that in which stage of prophase 1 does the crossing over take place? So the answer is during the Pachitine phase. The fourth stage of prophase 1 is called as Diplotine. Now during Diplotine, synapsis ends. So now the exchange of genetic material has already taken place. And synapsis ends with disappearance of synaptonemal complex. Homologous pairs remain attached at chiasmata. They begin to repel each other except at the point of crossing over. So here you can see that gradually the synaptonemal complex has disappeared. The place where they were the two homologous chromosomes had come in contact, that was synapsis. So that has ended, and synaptonemal complex has also disappeared. But still you find that the homologous chromosomes are in contact at the place of chiasmata, that is the place where the crossing over has occurred. They remain still attached to one another. They only begin to repel at the places other than where the crossing over had occurred. The fifth and the last stage of prophase 1 is called as dikinesis. In dikinesis, chromosomes become fully condensed. That is, now they are very clearly visible. They have become short and thick, and they are clearly seen. And the nuclear membrane and nucleus completely disappear. So the nuclear membrane, nucleus has completely disappeared. It looks like an X-shaped structure. So now here you can clearly see that the prophase has ended, nuclear membrane has disintegrated, that is, it has disappeared completely, and you can see that the structure is now looking like X-shaped, and still you can see that the homologous chromosomes are in contact with one another only at the place of crossing over. Apart from there, they have started repelling each other. So with this, we come to the end of prophase 1 of meiosis 1, and it had got 5 stages to it. We will now see what changes take place during the metaphase 1 of meiosis,